muscles contract, and there is movement. And fundamental to movement is the single muscle fiber, the basic unit of contraction. When a fiber of a voluntary muscle is examined under the light microscope, a definite pattern of light and dark stripes, or striations, is observed. During contraction, there is an obvious shortening of the entire fiber. On more detailed examination, however, this contraction is seen to be the result of the much finer shortening of the striations themselves. The substructures responsible for this movement could not be adequately observed until the development of the electron microscope. Then the complex internal arrangement of the muscle fiber was revealed. It was found that the fiber is made up of small substructures, fibrils, and the fibrils in turn are composed of still smaller units, which were named filaments. Two kinds of filaments could be distinguished: relatively thin ones which form the light stripes, and relatively thick ones which form the dark stripes. This accounts for the striated appearance of the fibril. Examination of a fibril in cross-section showed that the component filaments form a definite pattern. This pattern is due to the regular arrangement of the thick and thin filaments. The thick filaments, on chemical analysis, were found to be composed of the protein myosin, while the thin filaments are composed of the protein actin. Based on the visual evidence of electron microscope studies, a working model of how the filaments behave during contraction was constructed. It suggests that during contraction, the filaments slide over and past one another. This is the sliding filament theory of muscular contraction. The chemical interactions of the thick and thin filaments during contraction has been intensively studied by Dr. Georges Maréchal of the University of Louvain, Belgium.
The visual evidence for the sliding filament theory is quite strong. But in order to accept it completely, we must have additional evidence that these filaments can contract. If they cannot, then the theory is without any value whatsoever. In order to test this theory, we are making two preparations of the same voluntary muscle. From the first preparation, we are extracting all the chemical compounds of the muscle except the filaments. The muscle juice contains a number of chemicals, including the compound adenosine triphosphate. From the second preparation, we extract only the filament materials. From the thick filament, we get the protein myosin. From the thin filament, we get the actin. In the process of extraction, the myosin and actin combines to form a substance called actomyosin. Here is a solution of actomyosin. Now I'll squirt the solution into the water, and I get a beautiful thread. Now the question is to know whether this thread can contract. As we know that muscle can be quite easily stimulated by electrical shocks, let us try first this procedure. No response. Try again. No contraction. The thread cannot be excited with electrical stimuli. But it can perhaps be contracted with chemical stimulus extracted from the muscle itself. Let us try. It contracts beautifully So this experiment shows us two things. First, that the actomyosin is really the contractile proteins of the muscle. And second, that the nerve impulse somehow causes the release of some chemical factors which, when it is brought into contact with the actomyosin, causes its contraction. The question now is to identify this factor. Here I have attached to this rod a muscle from a rabbit which has been dipped into glycerol in order to remove all the components of the muscle cells except the filaments. Let us take one of these filaments of the muscle. Well, this one is a bit too small. Here is a beautiful one. Now here I have several substances which have been isolated from the muscle. And we'll try to make the thread contract by adding them one at a time. Let us try first creatine phosphate. Does not contract. Let us try lactic acid. Nothing. Phosphate. You find many phosphate in the muscle. Nothing. No contraction. Let us try another substance, calcium, for instance. Nothing. The cells are known to contain a lot of ATP, adenosine triphosphate. It contracts beautifully. So it appears that ATP is the key substance which is necessary for the muscle contraction. Let us try another experiment on a fresh fibers in a fresh solution. And now let us add the ATP. There is no contraction. So it appears that in muscle, the situation is not quite as simple as we thought. Besides ATP, some other component is also needed. Let us try again to add the other substance one at a time. Creatine. Let us try now with lactic acid. Nothing. Calcium. There is beautiful contraction. So these experiments show that ATP and calcium both are needed together in order to trigger the contraction. For a long time we wondered what happened inside the muscle. But now we think that we can have a clear description of the chain of events that is occurring. Let me show you on a simple model what happens. Now here we have the thick filament of myosin and the thin filament of actin. Now I need a muscle cell. It can be represented by this paper. In the muscle cell there are the thick filament of myosin and the thin filament of actin. The calcium in the resting muscle is not in contact with the filament, but it is kept inside some membranous structure that I can represent with this bottle. Now we have another membranous structure which are represented by these magnets all along the filaments. And in the resting muscle we have also the adenosine triphosphate with its 3 phosphates. This adenosine triphosphate is in contact with the filament. Here is the nerve which innervates the muscle cells. If a nerve impulse comes along this nerve, it will trigger an electrical disturbance which will travel along the membrane of the muscle. This electrical disturbance triggers the secretion of calcium ions which will reach all the filaments. This initiates the contraction. The energy for the contraction is furnished by the ATP. The last phosphate of this ATP is broken down and the energy released is used to bring about the contraction. Then, at the end of the contraction, the calcium is removed, and because the calcium is gone, the contraction stops. The ATP is then resynthesized, a new terminal phosphate is added, and the muscle has returned to its resting conditions. It is then ready for a new contraction. But of course, things are not so simple. The chemical reactions are vastly more complex. But in a simple way, I have demonstrated the basic events which occur during the contraction and how the energy of ATP is used to produce the muscular contraction.